Good morning to all. Welcome to the Diamond Group of Institutions Neet and K-Cet online classes. Yesterday we have completed molecular base of inheritance chapter. From that, still some of the important and previous questions we have to discuss. Continuation of this yesterday's class in molecular base of inheritance. First question: A DNA is. See here, DNA is a genetic material. So we are going to classify the DNA based upon the number of nucleotides per turn and the diameter of the two strands and helix of the DNA. It was classified into 5 types. So those are the 5 types of the DNA are aDNA, bDNA, cDNA, ddNA and zDNA. So question is asking in the form of aDNA. See now, all of the DNA is A, B, C, D. These all are their present in the form of right-handed foldings, except that zDNA is present in the form of left-handed foldings. And other major differences between the aDNA and the zDNA, otherwise A, B, C, D DNA and the zDNAs are in between. The major difference between the A, B, C, D and the zDNAs are number of nucleotide base pairs per turn. So the number of nucleotide base pairs per the turn, aDNA it's contained as 11 nucleotide base pairs per the turn. bDNA we know it's a standard DNA, it's contained as 10 base pairs per turn. And the cDNA it's contained as 9 base pairs per turn. And ddNA it's contained as 8 base pairs per turn, and the zDNA only it's contained as 12 base pairs per turn. Each one of the helix or coil or turn it's contained as aDNA as 11 base pairs, bDNA as 10 base pairs, cDNA as a 9 base pairs, ddNA 8 base pairs, zDNA it's contained as a 12 base pairs per turn or helix. So now you see aDNA is option they given as left-handed helix with 12 nucleotide base pairs. So here 12 nucleotide base pairs and left-handed it's something as a zDNA. A, B, C, D DNAs all are there present in the form of right-handed foldings, and the zDNA only it was present in the form of left-handed folding. And here aDNA that is option A, left-handed helix with 12 nucleotides. Left-handed helix with 12 nucleotides is transferring as a zDNA, not as aDNA. Right-handed helix with 11 base pairs per turn. So aDNA can show the right-handed helix and it's contained as 11 base pairs per turn. So option B is the correct one. And C, they given as right-handed with 12 nucleotides. So here 12 nucleotides it belongs into the zDNA. And left-handed with 11 nucleotides. Here left-handed DNA it's one only, that is the zDNA. So answer is B. The next question, in a 3.2 kbp it's represented as kilo base pairs long piece of DNA, 820 adenine bases were found. What would be the number of cytosine bases? So these type of questions will be asked in your objective exams in the Neet and K-Cet. So now you see they given as a hint, it is a segment of the length of the DNA, it is 3.2 kilo base pairs. So here kilo base pairs means we have to represent it in we can considering as 1000. So 3.2 into 1000 will be get as a 3200 base pairs. So if the length of the DNA it's contained as a 3200 base pairs. The length of the DNA is contained as a 3200 base pairs. Out of the 3200 base pairs of the DNA, 820 adenine bases were found. So 820 adenine bases are found means you know the charge of rules. Adenine is pairs with the timing. Number of adenine equals to number of timing. So 820 adenine means equals to 820 we are going to represent it as a timing. And so here 820 adenine, 820 timing. and the question was asking as a how many number of cytosine are present. How many number of cytosine molecules are there present? See now, the length of the DNA it is 3200 base pairs. And out of the 3200 base pairs, they given as a hint it is 820 adenine molecules. 820 adenine molecules means 820 thymine molecules. So 820 plus 820, 1640. So when we done 320 minus 1640 will be get into the cytosine and guanine. So number of cytosine equals to number of guanine. So 320, 3200 minus 1640 will be get as a 14. 1640. One minute, sir. So will be get as a 320 minus 1640 will be get as a 15,000 1560. So 1560 equals to guanine and cytosine. So applying of the Chargaff rule, number of purines are equals into the number of pyrimidines. So number of purines are equals into the number of pyrimidines. Number of cytosines are equals into the number of guanines. These both are will be get as a by combined together we got into the 1560. So number of cytosine molecules only you want. So that's why by 2. So now here two two sevens and two eights 780 molecules means out of the 1560 molecules 780 molecules are guanine and 780 molecules are cytosine. So number of the cytosine molecules are there asked, so that's why number of the cytosine molecules are 780. So here the twist of the question is length of the DNA was not given directly. Length of the DNA is given in the form of kilo base pairs. So the 3.2 kilo base pairs are we have converted into the base pairs, that is by the multiplying with 1000 because kilo means 1000. So now we'll be get as a total number of the base pairs are 3200, and out of the 3200 adenine and thymine base pairs are adenine thymine nucleotides or base pairs are 1820 base pairs. Those are represented as a 1640. So these are the 1640. We are going to equals to adenine and thymine. We are going to reducing the adenine thymine molecules, subtracting of the adenine and thymine molecules in the 3200 base pairs will be get as a 1560, and these are the 1560s contain as a guanine and cytosine. We are going to divide into will be get as a 780 molecules. The 780 it is cytosine and guanine. Question asked as a cytosine, so that's why number of the cytosine molecules in a 3.2 kilo base pairs it is 780. And the next question, if there are 20 adenine molecules in a bDNA, see now 20 adenine molecules in a bDNA, double structure showing 20 coils. So here you want to know bDNA, it's called as a balanced DNA, it's also called as that is stable DNA. That bDNA is right-handed fold length, and bDNA each turn or each helix or each coil is contained as a 10 base pairs. It is equals to 3.4 nanometers. 3.4 nanometers. It is equals to 20 bases. So now if there are 120 adenine molecules in a bDNA, double helical structure showing as a 20 coils, what would be the number of pyrimidine nucleotides forming 3 hydrogen bonds in it. So pyrimidines forming 3 hydrogen bonds in it, that is 3 hydrogen bonds means we are going to depend as cytosine and guanine. So now out of that cytosine and guanine, purines and pyrimidines, they are asking now the pyrimidines, so we have to identify again the cytosine molecules. That hint it is 120 adenine molecules in a 20 coils. So 20 coils represent as here each one of the coil is contained as a 10 base pairs or 20 bases. So we can multiply into 20 into 20 will be that as a 400 bases. Out of the 400 bases, adenine is a 20. So adenine is 20 means thymine also 120. So number of adenine and thymine molecules are 240. So minus 240 will be that as a 160 will be there as a 160. They are equals to cytosine and guanine. So number of the cytosine molecule they are asked that a 3 hydrogen bonds contain as a guanine and cytosine only. So you want to know only the pyrimidine. Purines are adenine and guanine. Pyrimidines are cytosine and thymine. 160 by 2 will be that as a 160 by 2 will be that as a 80. So answer it is 80 molecules. Number of cytosine molecules as a 80 in a 120 adenine molecules of 20 toils DNA. And the next question, when DNA replication starts. So DNA replication always we are going to depend as mostly in the bacteria and the eukaryotic organisms has a semi-conservative mode. The semi-conservative mode of the DNA replication it is one strand that we can take as a segment of the DNA. One strand of the DNA came from the parent, another one of the strand of the DNA is the newly synthesized. So when DNA replication starts, where DNA replication starts? So options are leading strand produces autozygotic fragments. So this is the replication force and 5 prime to 3 prime direction, and this is the 5 prime to 3 prime. So this strand is called as leading strand and this strand is called as lagging strand. So when DNA replication starts, leading strand produces autozygotic fragments. So leading strand will not produce the autozygotic fragments. So option A in that statement, lagging strand only can produce the autozygotic fragments. The hydrogen bonds between adjacent of two nucleotides of the two strands. So now here the hydrogen bonds between the two adjacent of the two adjacent nucleotide of the two strands break. So here when we seen into the replication fork, here the hydrogen bonds will be going to present. Always DNA replication will going to start whenever the hydrogen bonds will be going to break down means from here. So the hydrogen bonds between the two adjacent nucleotides, these are the two nucleotides, strands breakdown of the two hydrogen bonds in between the two strands of the DNA. So from here onwards, always DNA replication will be going to start. So answer it is B. And option C, 1C, the phosphodiester bonds between the adjacent nucleotides. So you can see as a phosphodiester bonds between the two adjacent nucleotides. So nucleotides means nitrogen base, sugar and the phosphate molecule. This is the nitrogen base and the sugar and phosphate molecules. So this is we are called as phosphodiester bonds. So here DNA replication will not start in between the sugar and phosphate molecules. DNA replication always starts from the two strands of the DNA where the hydrogen bonds will be going to break down. In between the two strands of the DNA, hydrogen bonds are break down. They can form into small loop-like structures. From there onwards, DNA replication will don't start. So phosphodiester bonds between the two adjacent nucleotides, it is worked by the restriction endonucleases. Restriction endonuclease will be drawn to work to the breakdown of the DNA, that breakdown of the DNA in between the sugar and phosphate molecules. And option D, the bonds between nitrogen base and the deoxyribose sugar break. So in between the ribose sugar and the nitrogen base, this is called as glycoside bond. So in between these, the breakdown of by the breakdown of the glycoside bond and by the breakdown of the phosphodiester bond, DNA replication will not start. Replication always will don't start by the converting of the double-stranded DNA into a single-stranded. So converting of the double-stranded DNA into a single-stranded, in that regions, double-stranded, the two strands of DNA is closely connected together with the presence of hydrogen bonds. By the breakdown of the hydrogen bonds only, DNA replication will start. So that's the answer is B. The next question. In prokaryotes, the process of replication catalyzed by the following enzymes. Identify which of the enzymes coordinate with role. which are the enzymes but coordinate with the role. See now, they're given as a helicase enzyme. Helicase enzyme, they're given as a function joins the ends of DNA. Helicase enzyme, its function, it is called as a its function of this helicase enzyme. At the time of DNA replication, two strands of the DNA will be connected together with the hydrogen bonds. That hydrogen bonds will going to break down with the helicase enzyme. But the function it is function that mentioned as join the ends of DNA. Join the ends of the DNA, it can uttered by the lydase enzyme, not the helicase enzyme. It's an incorrect match. And DNA polymerase 1 and polymerase 2. Polymerase 1 synthesis the DNA. So DNA polymerase 1, polymerase 2 and polymerase 3, three types of the DNA polymerases are involved into the DNA replication. Out of the three types of the DNA polymerase 1 and the polymerase 3, they are majorly involved into the DNA replication process. DNA polymerase 2, its function is not mentioned, and DNA polymerase 1 and polymerase 2, both of the enzymes will involve into the proofreading mechanism. So, but here they given as DNA polymerase 1, synthesis of DNA polymerase 2, erase the primers and fill the gaps. So, filling of the gaps is the function of repairing only. So, DNA repair is maintained by the polymerase 1 and polymerase 2. Polymerase 1 is not synthesized the DNA. Majorly DNA synthesis takes place by the involvement of DNA polymerase 3. And option D they given as a primase. So, the primase enzyme function synthesis of RNA primers. At the time of DNA replication, after the formation of single stranded RNAs, always into the 5' strands, RNA primers are added. That RNA primers are synthesized by the primase enzyme. So correct match for this, and its role is D. Primase synthesis of the RNA molecules, RNA primers. Yes. Next question. In a given segment ATG, ACC, AGG, ACC, CCA, ACA is a first base get mutated. The effect of this uncoding by DNA segment will result in. So they given as a one segment of the DNA. So in these are the segment of the DNA. This is the sequence. So out of the segment of the DNA, first segment of the DNA will be going to mutated. Mutation means change the gene sequence. If the segment of the DNA of first nucleotide, it will be going to mutated, what will be going to happen? So this is the DNA segment, means the DNA sequence. The DNA sequence, 3 nitrogen bases are we are going to represent as a 1 codon. So like that arrangement of the DNA nitrogen bases are present in a DNA. So now if you are going to change the first nitrogen base, so if you are going to change the first nitrogen base, or if you are going to mutate the first nitrogen base, the effect on the coding by DNA segment will result in. The effect of the coding segment of the DNA, it is options of complete change the type as well as a sequence of amino acids, change in the first amino acid only, no change in the sequence, one amino acid less interruption. See now the concept of these replication, transcription and translation. Here 2 strands of the DNA, 1 strand of the DNA 3' to 5' and 5' to 3' direction. So these are the double strand DNA. Out of the 1 strand of the DNA, out of the 2 strands, 1 strand of the DNA always we call as a template strand. It can involve into the synthesis of mRNA. This strand will be don't involve in the synthesis of mRNA. This is called as a transcription 5' to 3' direction. So this is the mRNA. So now this mRNA can undergoing as a translation mechanism that can synthesize the polypeptide chain. It's a protein. So now here the DNA contain as a segment of nitrogen bases of these A, T, G, and the nitrogen base are present. Out of the these nitrogen bases of the segment of the DNA, first nitrogen base will be going to mutated. What will be going to happen? If you are going to change the first nitrogen base, So what will be going to happen is the complete change the type as well as the sequence of amino acids. So here complete changing of the DNA sequence and the sequence of the amino acid pairing will not change because here it will contain as a ATG. So this A sequence A it will be done mutated into the C. So these are the nitrogen base contain of the proton will be don't change. That proton will be don't code with the one particular amino acid. If the nitrogen base will be don't change, automatically amino acid also will be don't change. So that entire sequence of the DNA and entire amino acid sequence will not change. And option B, change in the first amino acid. See now, here you are going to change in the first nitrogen base of the proton. Instead of the A, you are added into a C. It will be drawn to form as, it will be drawn to CUC will be drawn code into the one amino acid. So these are the CUC coding of the amino acid and UAC or UAG will be drawn code. It is a nonsense codon. Instead of the nonsense codon, CUC will be drawn to presence. Amino acid will be drawn to change. First amino acid only will be drawn to change. Remain all of the amino acids or remain all of the protons on mRNA and the DNA sequences will be drawn to same only. No change in the sequence. One amino acid is less interrupted. So one amino acid less in the protein. One amino acid less in the protein will not take place here because we are going to change in of the DNA. We are going to change in of the proton of nitrogen base. So by the changing of single nitrogen base, single amino acid sequence or single amino acid will be don't change. Remain all of the protons and remain all of the amino acids are present in same only. Yes. See the next question. Central dogma was proposed by. So here central dogma of molecular biology represented as a flow of genetic material DNA to RNA, RNA to protein. So this is the flow of genetic material or the central dogma of molecular biology. So that central dogma of molecular biology is proposed by options are given as a Twitt, Biddle and Tatum, Temin and Baltimore and Flood. See now here, Twitt, he is a scientist. He was proposed the flow of genetic material in a one way or one side direction, that is the DNA to RNA, RNA to protein. DNA to RNA it's called as a transcription, RNA to protein it's called as a translation mechanism. So central dogma of molecular biology was proposed by Frick. And option B, Biddle and Tatum. He is a scientist was proposed by one gene, one enzyme mechanism. And option C, Temin and Baltimore. So these are the two scientists that can propose the Teminism. These are also called as Teminism, are also called as reverse transcriptase. Reverse transcriptase. So Teminism or reverse transcriptase mechanism says as synthesis of the DNA from mRNA. This is we call as a reverse transcription process. That reverse transcriptase mechanism or reverse transcription mechanism or central dogma of molecular biology is reverse proposed by the Temin and Baltimore. And Flood, he was proposed the tRNA. It was present in the form of L-shaped structure of the tRNA. So central dogma of molecular biology is proposed by the Twitt. Central dogma of molecular biology reverse it was proposed by the Temin and Baltimore. So it is also called as Teminism. Question was asking as a central dogma of molecular biology only. So answer is A. If the question was asked as a central dogma of molecular biology is reversed, that at the time will be that as a answer C. And the next question, which amino acids are present in histone? So when we seen as a nucleosome concept, the DNA always presents in a negatively charged. That negatively charged DNA is wrapped into the positively charged histone protein. These are the histone automa, or we are also called as histone proteins. So DNA it was present in negative charge because of the presence of the phosphorus. And histone atomers are they contain as a positive charge due to the presence of two amino acid or positive amino acids. They given as lysine histidine, valine histidine, arginine and lysine, and the arginine and histidine. So due to the presence of lysine and arginine amino acids, these are the histone proteins are will be rotated into the positively charged. The natively charged DNA wrapped on the surface of the positively charged histone protein, it's called as a nucleosome. So the histone proteins are they having positive charge, positive nature due to the presence of two amino acids as a arginine and lysine. The next question, the pioneer contribution towards the human DNA fingerprinting, forensic science India. So the pioneer contribution towards the human DNA fingerprinting forensic science in India is, that is, DNA fingerprinting was proposed in India is, options are given as Lalji, Watson and Trick, Swaminathan and J.C. Bose. So Dr. Kashyap and Lalji Singh. So these are the two scientists, the father of DNA fingerprinting in India. So these are two scientists they proposed. These are the two scientists they proposed the DNA fingerprinting in India. That is, those are the scientists are Lalji Singh and Kashyap. And then what about the Watson and Crick? These are the two scientists, Watson and Crick, that can explain the structure of DNA. And Swaminathan is the father of green revolution. And J.C. Bose, Jagdish Chandra Bose, he was proposed the crystallograph. By the using of the crystallograph, plant length will be done to measure for the fraction of seconds. So DNA fingerprinting initiated in India by the Kashyap and the Lalji Singh. And next question, match the following: VNTRs, introns and exons, dystrophin, satellite. And largest gene, DNA fingerprinting, bulk DNA and splicing. See now, VNTRs, variable number of tandem repeats. Variable number of tandem repeats, we are going to use as a VNTR sequence for DNA fingerprinting. Introns and exons. So introns and exons are removal of introns and addition of exons. Only it's called as a splicing mechanism. Dystrophin is the largest gene, and satellite DNA we are called as a bulk DNA. The satellite DNA is separated from the bulk DNA. So VNTR faced with the Q DNA fingerprinting AQ, and introns and exons BS. AQ and BS, answer is B. Next question. Which of the following is not required for any of the techniques of DNA fingerprinting available? So options are PCR technique, zinc finger analysis, restriction endonuclease, and DNA RNA hybridization. And here DNA and DNA hybridization mechanism. So DNA fingerprinting is used to detecting of the criminals by the using specialized mechanism or specialized structures we call as VNTR sequences in the human beings. VNTR sequences, every human being, 99% of the DNA will be done similar, but 1% of the DNA will be different. That 1% of the DNA only we call as VNTR, variable number tandem repeats. So based upon the VNTR sequences, we can easily identify the criminals. So by the process of DNA fingerprinting. In this of the DNA fingerprinting mechanism, we are going to use as some of the equipment, those are PCR techniques. Polymerization chain reaction. It is used into the amplification of isolated DNA or amplification of the DNA. And option B, zinc finger analysis. So here, zinc finger analysis is not used in a DNA fingerprinting. And option C, restriction endonucleases. These enzymes are used into the cleaving of DNA at specific sites. These are also used into the DNA fingerprinting. And DNA hybridization mechanism. We are going to using as a DNA primers. So these are the DNA primers they will be going to present in the form of single stranded, and they can bind into the specific sites of the single stranded DNA, that is called as DNA hybridization. So it is called as a southern blotting technique. So in a DNA fingerprinting, PCR equipments and restriction endonucleases and the southern blotting techniques we are going to use. Zinc finger analysis is not related into the DNA fingerprinting. So answer is Z. The next question, which of the following factors RNA polymerase transiently to initiate the transcription? So when we seen into the transcription mechanism, 3 prime to 5 prime, 5 prime to 3 prime, these are two strands of the DNA. Always transcription mechanism will going to take place by the involvement of one strand only. That strand we call as a non-coding strand. That non-coding strand is having as a polarity 3 prime to 5 prime direction. So always synthesis of the mRNA from the DNA, that is taught from the involvement of different types of the factors. The different types of the factors are initiation factors. That initiation factors we want to start the synthesis of by the involvement of. Options are they given as a rho factor, beta, gamma, and sigma factors. So transcription unit, it's contained as a promoter, structural gene, or we are also called as initiation, elongation, and termination. So in that initiation start the synthesis of enzyme, synthesis of mRNA from the DNA by the involvement of initiation factor. That initiation factor it is a sigma factor. So sigma factor is the initiation factor. Rho factor we call as a terminating factor. In these, by binding of the sigma factor in the region of promoter, start the synthesis of mRNA will take place. So answer it is sigma factor D.